As late as 1951, the chemical structure of DNA remained a mystery. At Cambridge University, an American biologist, James Watson, and British physicist Francis Crick were working to solve the mystery of the structure of DNA. Some facts about DNA were already known. For example, scientists knew that DNA was composed of 4 bases: adenine, thymine, guanine, and cytosine. A paired with T and G paired with C. However, no one knew how these pairs connected to one another. Knowing the structure of DNA would open the door to understand how information travels in living systems. Not surprisingly, Watson and Crick were not alone in their attempt to discover the structure of DNA. At King's College in London, physicist Maurice Wilkins and chemist Rosalind Franklin were attempting to unlock the mysteries of DNA structure. Rosalind Franklin was an expert in X-ray crystallography, a technique involving an X-ray beam traveling through a crystallized DNA molecule and capturing a shadowy image of its internal structure on a photographic plate. Rosalind was able to capture one of the most valuable images in science, which she called Photo 51. It was with this photo that Watson and Crick were able to determine that DNA was a double helix. However, the photo was given to them by Franklin's colleague, Maurice Wilkins, without her knowledge or consent. This last piece of the puzzle enabled Watson and Crick to compose a 3D model of the structure of DNA. In 1962, they, along with Maurice Wilkins, won the Nobel Prize for Physiology or Medicine for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. The discovery of the structure of DNA sparked a scientific revolution. It brought to light the molecular and biochemical information of life and opened doors to other areas of research that was once thought to be impossible. Sadly, and some would argue unfairly, Rosalind Franklin was not considered for the 62 prize. The rules state that it can only be awarded to a living recipient. Franklin died in 1958 of ovarian cancer, likely caused by her exposure to X-rays. Her contribution to the discovery of the structure of DNA is not forgotten.